Good afternoon, everyone. The Atoms Group is pleased to have with us today Professor Pedro de Alcântara Pessôa Filho. He is currently a full professor at the Chemical Engineering Department at the Polytechnic School at the University of São Paulo, Brazil. He obtained his undergraduate, master, and PhD degree in the State University of Campinas. Among his research interests, we have phase equilibrium thermodynamics, with an emphasis on the study of biomolecules, purification process, but also with application in other branches of knowledge, such as petroleum and food engineering. In the field of food engineering, he is a member of the University of São Paulo Center for Research in Food and Nutrition. He is also an associate researcher at Food Research Center, one of the Centers for Innovation, Research, and Dissemination, supported by Prof. Professor Pessôa has published over 70 articles in international journals, which to date have been cited more than 600 times. He is also a member of the editorial board for the Brazilian Journal of Chemical Engineering. So welcome, Professor Pessôa. We're very pleased to have you here, and I'll have the word.
Thank you, Yamara, for the so kind introduction. And I would like to start by thanking all the Atoms Group for the invitation to present this lecture today. I'm really very glad to be here. Of course, I would prefer to be personally there to shake hands and hug everyone, but these are hard times for everyone. So I will share my screen here. Okay, can you see it?
Yes.
That's okay. Oh, great.
That's great.
So the topic of the lecture today will be Thermodynamics of Biomolecule Downstream Processing from Bench to Batch. So a short outline of the presentation. I will have a very short introduction on the relationship between thermodynamics and biomolecules, and I will try to answer three main questions. How do the experimental data look like when you are talking about mainly on solid-liquid equilibrium in bioprocessing? How can we relate phase equilibrium and biological activity? And how can we extend the research for industrial process? So the first question is thermodynamics and biomolecules. What these two words have to do one with the other? Well, some of you, perhaps all of you, will remember that one of the first statements of the first law of thermodynamics was due to Julius Robert Mayer. He was not a physicist, but he was a physician. And he observed that movement, temperature maintenance, and metabolism are connected. So to give you an example of this reasoning, I selected here a hamster, for example. We know that this hamster gets energy from a single source, which is food, and uses this energy both to maintain its body temperature and to do work. Well, some of you have already seen these slides, and I hope that those who didn't see it are smiling behind the Zoom cameras. But I somehow misguided you. I will not talk about biothermodynamics, which is really the energy change study in living systems. And I will not talk about biochemical thermodynamics, which is the study of equilibrium, entropy, and Gibbs energy changes in biochemical reactions. These are very well established research fields, but their concepts are not commonly used when we are working with biomolecule downstream processing. So, what will we talk about today? Really about unit operations for biomolecule separation and purification in bioprocess, which are somehow similar to downstream operations usual in chemical industry. We have such as we have precipitation when we are producing, for example, a salt. We have precipitation when we are producing and antibiotics, a protein, an enzyme, many other molecules. So we are really talking about unit operations, usual for the chemical engineering processing, but directed to the bioprocess. We have here, which is a kind of classical bioprocess. Of course, it's difficult to see anything here. It's just small letters all over the slide, but this is an overall scheme of the production of insulin, which is a kind of classical bioprocess in the sense that insulin was the first biomolecule to be produced in industrial scale using a genetically modified microorganism. So what we have here, we have pretreatment steps, the steps that precede the bioreaction. The bioreaction itself, and here we have the downstream processing, which shows us that really the downstream process may be the most important part, not the most important part, but a very significant part. All parts are important in a bioprocess. There is no rule of thumb on the share of the costs which are associated to downstream operations, but they may be up to 80–90% for therapeutical proteins such as insulin and monoclonal antibodies. Of course, for bioethanol, this is not a high share. It's a biomolecule also, but for insulin and monoclonal antibodies, for example, they may be up to 90% of the overall production costs. This is a node reference, but these values didn't change that much in the last 10 years. How is the general structure of downstream process? We can recognize 4 major steps when we leave the bioreactor until we get to the final product. We have what we call primary recovery operations, cell harvesting, cell cleavage, clarification. We have then low resolution operations, which are operations that aim at mainly at concentrating the biomolecule. So precipitation, liquid-liquid extraction. We have then high resolution operations, which are mainly chromatographic steps, and they really aim at purifying the biomolecule. And we have final disposal operations, which are operations that aim to put the biomolecule in such a state that it can endure shelf life. Here, we, as mostly thermodynamicists, we can recognize equilibrium operations, and here, solid-liquid equilibrium operations, which will be the focus of this lecture. Well, what else makes the downstream processing different from the conventional separation unit operations in chemical engineering? We have to take care of biological activity. It is essential. It's just as essential as purity. If I get a pure compound in the end, but inactive, I get nothing. The phase behavior is somehow more sensitive to system conditions. It is whimsical and in many places counterintuitive. And the experimental uncertainty is also higher than for conventional systems. When we measure protein concentration with 10% of experimental uncertainty, we are sometimes happy with that. And of course, it's a very large uncertainty for other systems. So the first question that I want to answer here is, how do the experimental data look like? So I will address the main aspects that we have to consider when we are dealing with phase equilibrium on precipitation and crystallization. What is solubility? What changes the solubility? How is the structure of solid phase? It can be an amorphous solid, it can be a crystal phase, and how do these things related to auxiliary measurements and analysis. So here, we will explore a comprehensive set of experimental data for a single protein with a single salt. So here we have a picture with the solubility of lysozyme, which is an enzyme, in sodium succinate solutions at a fixed pH and a fixed temperature. We often achieve this phase separation, this precipitation, by adding a third component. It may be a salt, it may be a polymer, a polyelectrolyte, an organic solvent. There are many substances that are used for that. We see here the solubility as a function of the ionic strength, which is a measure of the salt concentration. Both measurements are actually related to what we call supernatant, which is the liquid phase, which is in equilibrium with the solid phase. This kind of data is often modeled using the Kohn equation. It's a very simple equation, and you can see here that the logarithm of the solubility is related to the ionic strength. If we change any aspect, anything here, temperature, pH, salt, we get different diagrams. For example, here is a comparison with sodium tartrate at almost the same pH, the same temperature, the same protein, and we get a completely different phase behavior, even though we can model using the same Kohn equation. Well, looks simple, solubility data. Yes, but there are some tricks in that. Let's get here we have a typical kinetic experiment concerning the determination of solubility. What we have here, we have the concentration in supernatant as a function of time. It's a typical point from one of the points that I presented before. What we can see here is that for almost 24 hours, we get no precipitation at all. These measurements here are simply experimental deviations, that's small changes in concentration. Then we have, it's not exactly that, but the induction time. We have a sudden drop in protein concentration. So the protein starts to precipitate, but it may take up to 1 week for equilibrium. Here it's less than 1 week, but we often wait until 1 week to have an equilibrium point, to be sure that the system is in equilibrium. So, but that's not everything. We have some auxiliary measurements, auxiliary parameters that help us to understand this phase equilibrium. One of them is what is called osmotic second virial coefficient. What is that? If I write the osmotic pressure as a function of protein concentration, the second virial coefficient is simply this term here. It depends on the salt concentration, on temperature, on pH. It can be obtained through different experimental methods, osmotic pressure determination, but also through light scattering, through self-interaction chromatography, to sedimentation experiments. This value is related to the outcome of the operation. If I get an amorphous solid or a crystal solid, the second virial coefficient somehow is a guide to see what solids I will obtain in this case. So let's see, here we have a set of data, also for lysozyme in sodium succinate solutions. These data were obtained through self-interaction chromatography. And here we have the so-called crystallization window or crystallization slot. This is the range of values of B22 for which I expect to get a crystal phase. So If the value is higher than that, I will probably get no precipitation at all. If the value is lower than that for high ionic strength, I will probably get a amorphous precipitate. So let's investigate that. Here we have a map of different situations that we can investigate for determining where we really get crystals. So we have here the supersaturation ratio. That's the ratio from the initial protein concentration to the solubility. So I can vary this supersaturation ratio and vary the ionic strength and see when I get crystal phases, solid crystal phases. And actually, of course, we choose carefully the data, so everything here is within the crystallization slot. And indeed, we got crystals at those points. It's not that we got amorphous phase in the other ones. We just didn't get precipitation because the kinetics was too slow in this case. Well, but this value of B22 is a guide. Is that really the whole truth? Let's investigate. So we made another experiment. Each point here is a different experiment. We extended the data for ionic strengths where I didn't expect a crystal and for higher supersaturation ratios. And what we got here is crystals all over these points. And actually here, the biggest crystals were obtained in this range here. And they are really very, very beautiful. This photograph is here. I almost printed it and hanged it on the wall because it's really, really beautiful. So in this first part of the lecture, I've shown how the experimental data look like. We explored the relationship between solubility, the kinetic hindrance, the second virial coefficient, supersaturation, and the solid phase structure. And the cautionary conclusion is that general rules are important guides, but they are not the universal truth. The second question is, how can we relate phase equilibrium and biological activity? Perhaps the question should be, can we do that at all? Is it possible? Well, here we have to take care that the protein is actually concentrated in the solid phase. So we have to investigate deeper the solid phase and the solid liquid equilibrium beyond the solubility. So what we did here? Here we have a phase diagram. These are overall compositions for phase equilibrium investigating. The diagram is somehow upside down. Salt and protein concentrations are inverted, but there is a reason for that. So we mix it, also lysozyme with ammonium sulfate and water at a fixed pH, a fixed temperature. So we just mix it, a salt solution with a get these overall concentrations here. We wait and get a phase separation. We can measure and plot in this diagram the supernatant compositions. That means the compositions of the liquid phase, just as we did. But we can also measure the composition of the solid phase, of the precipitate. Actually, these compositions are of the precipitate, but are not the composition of the solid phase. And why not? Because the solid phase has some liquid phase with it. We cannot, for a protein system like that, it's difficult to wash, and we always have the liquid phase with the solid phase. So we are measuring actually a mixture of the solid phase and the liquid phase. We can use this data to infer the composition of the solid phase using a quite old technique, which is called, whoa, there was a problem here with my, don't know what happened, but it's Windows. So this is called Schleinemakers technique. So what we do, we extend the tie lines and we see if we get an invariant point. This invariant point will be the composition of the solid phase. This is an old technique. It's applied widely when analyzing phase diagrams of salts, for example, but we can do that for proteins also. So this would be the true precipitate, the composition, the real composition of the solid phase. Well, we get one invariant point here and a second invariant point here. We have two true precipitates. One of them is a protein hydrate at low salt concentrations, and the other one is a complex containing protein, salt, and water. Well, but if we can't just separate the solid phase, what's the importance of identifying these compositions? Well, if we analyze at the microscope, we can see here that when the precipitate is a complex containing salt and protein and water, we have a totally amorphous solid phase. There's no structure at all in this solid phase. But here we have crystalline structure. It's not as beautiful as it was, as I shown before for lysozyme in sodium succinate solutions. But anyway, these experiments were carried out without control of supersaturation, without any kind of intention of getting a crystal phase. It is surprising that we get some crystal phase here. But the most important thing is that the total activity recovery is different in both regions. So when the solid phase is a complex with salt, we get only 1/4 of the overall activity recovered in the end of the process. While when the solid phase is a protein hydrate, we get 90%, which is almost 100%, considering the experimental uncertainty here. Well, it might be due to the high salt concentration, the high ionic strength to which the protein molecule was exposed. It might be, but it's not. We investigated what happens with circular dichroism, fluorescence spectra, and there is no shift, no significant shift in these conditions. So there is no effect upon the tertiary structure of the proteins in these situations. Well, fine, but we have here just one situation. Yes, if you change temperature, for example, of course, we change the phase diagram. We change solubility, but we don't change the invariant points. So it's also an indication that these invariant points do represent the solid phase composition. And through many experiments with many salts at different temperatures, also at different pHs, I didn't show here, but we made some experiments at different pHs, we can see that for this system, for lysozyme and water and salt, we almost always get a solid phase, which is a protein hydrate, and sometimes we get a different solid phase, but it really can, this kind of experiment can be done and we can relate the phase diagram with the enzymatic activity recovery. So the answer to that question is yes, it can be done, but it's not an easy task. We must understand deeper what the solid phase is. And the bad news here is that it can't be done extensively. If I have a protein which is not high soluble in water, I simply can't conduct this kind of experiment. Okay, so I have shown how the experimental data look like and how we can relate somehow the phase equilibrium with enzymatic biological activity. So the last part of the lecture will be how can we extend the research for industrial process. So if I want to go to an industrial process and make this kind of thing significant, what I have to do? I have to understand the solid phase, understand its relation to the biological activity, understand the kinetics of the crystallization process, and then identify the key factors which affect the process and change the process within certified limits, within approved limits. So we'll consider here, I will show a case study with the industrial production of an antibiotic, such as virginimycin. Virginimycin, it is not a protein, but it's an antibiotic. It's used in veterinary medicine, mostly for cattle, produced by a microorganism, Streptomyces virginii. It was discovered in 1954 and produced since 1958. It's a good thing. It's an old molecule, and as such, it's safe. It's been used since then. It is produced in Brazil since 2006 by Fibro Animal Health Corporation. It was used formerly as a growth promotional agent, but this use has been phased out. Actually, virginimycin is an antibiotic, but it's a mixture of two main molecules. They are called factors. Actually, there are other factors in this mixture, but the main factors are called M1 and S1. So they are different molecules, similar but not equal. They are antibiotics themselves, but the important thing is that they have a synergistic effect. So if I make a plot here of the activity per biological activity per mass as a function of the composition, the fraction of one of these factors, I have a maximum point here at roughly 1/3 of S1 and 2/3 of M1. The industrial production of virginimycin has some typical, in some sense, steps. We have pretreatment steps, preparation of inoculum, sterilization, germination. We have the bioreaction itself. We have also primary recovery, clarification, liquid-liquid extraction with methyl isobutyl ketone. We have purification. Here is a crystallization operation that has two steps: solvent evaporation and anti-solvent addition. We have final disposal operations. We have to get rid of methyl isobutyl ketone and hexane, of course, because the antibiotics will be used after that. And we have solvent and anti-solvent recovery operations. They are separated through distillation. And we have also wastewater treatment. So our focus here will be on the purification steps. So what will be shown here? First, the characterization of the product. We have here the bulk virginimycin. So the analysis, the composition, the biological activity, crystal size, shape, structure, some thermal analysis will be presented for the substance to understand what are we dealing with. Then I will present some results on a lab scale process simulation in a fully instrumented workstation in which we can follow both the crystals, the crystal population, and the system, the liquid phase composition. And finally, we go to the industrial process improvement. Once we get the results from the lab scale simulation, we know what influences the crystallization, so we can go to the industrial process and see what we can change, avoiding incrustation, which is a very bad hazard. It's plastic, but to be avoided, and also respecting certification limits. So we got at first 4 samples from commercially available lots, no specific deviation, no process intercurrence. We analyzed the composition. The composition is presented as what is called the synergy factor, which is actually the factor that shows how much the mixture has, the activity of the mixture is higher than the activity of the separate factors. So it's a function of the fraction of one of these factors, M1 here, or S1, it's the same. And as I shown, we have a maximum around 2/3 of M1. The maximum value of F is 3.19. So here we have some results for these samples. And we can see here the activity is expressed as a percent of a standard assay. And we have the synergy factor. What we can see is that the first good thing, the synergy factor is pretty much close to the maximum. And that's very good. It indicates that the bioreactor is working properly. The strain of the Streptomyces virginis is a very good one. But what we can see here is the activity is not directly linked to the synergy factor. When we compare A1 and A3, we have the same activity, close synergy factor, but not the same. But which is even worse, when we compare A1 and A4, we have really the same synergy factor, but very different activities. So the synergy factor does not explain all the differences in biological activity. We can analyze the solid concerning the population of crystals. So there are many parameters that are involved in this characterization, but we have here the second moment, the third moment, and the cutoff diameter of half the population. And we see that samples are close one to another, but again, there is no relationship between this size distribution and the activity. When we compare A2 and A4, they have the same activity, but very different size, crystal size populations. So we went to analyze as much as we could the samples. So near infrared for the analysis of diffuse reflectance. It allows us a qualitative analysis in this case. We have it identical spectra, no significant differences. The only differences in baseline are actually due to the crystal differences in crystal size. Thermogravimetry also, we have similar thermal profiles. The thermal events are the same. The melting occurs around 150 degrees centigrade. It was determined through hot stage microscopy. Powder X-ray diffraction also, we have a crystal solid, it's not amorphous, totally amorphous, but no significant difference in crystallinity. Optical microscopy, hot stage microscopy, scanning electron microscopy, and differential scanning calorimetry also has shown us that there is no significant structural difference between samples. They're not equal, completely equal, but there is nothing that we can relate concerning the activity. So let's go on to see what happens in the process, but in laboratory here. So we have here the workstation that I've shown. It's a fully instrumented, we have two fully instrumented vessels here, and we can see the FBRM probe, which allows us to to follow what happens with the crystal population. We have a stirrer, the IR probe, which allows us to follow what's going on with the composition of the liquid phase. In this case, we use it as raw materials for the tests, what is obtained in the industrial process. So we started with a solution with methyl isobutyl ketone and the antibiotics and virginomycin, and it was obtained from the decantation tank. So there's a total solution mass here, the mass fraction, but it was obtained really from the industrial process. Hexane, which is the anti-solvent, is the hexane recovered by distillation. It's not 99.9% sigma Aldrich hexane. No. It's just the same hexane that is used in the industrial process. It's 99%. It's also quite pure, but not HPLC grade. The crystallization has two parts. This purification has two parts. First, we evaporate the solvent, methyl isobutyl ketone. It takes 3 hours and a half, approximately. It is performed under vacuum in a narrow pressure range and temperature 56 degrees centigrade. Then we have precipitation, the precipitation itself, which is carried out by adding hexane. After 2/3 of the methyl isobutyl ketone is evaporated, the addition time is 9 minutes. We do this hexane addition in trying to somehow mimetize what happens in the industrial process, but it's carried out at lower temperature, 28 degrees centigrade. The procedure can be seen here. So what's shown here in this picture? We have the stirring rate. It's maintained constant all over the process, except for when the hexane is added. We have the jacket temperature here, the medium temperature. It's kept constant in the evaporation step and also kept constant in the precipitation step. And we have here the hexane volume. Let's recall that we have here what we call the evaporation, and here the precipitation. The results, the outcome of the simulation is something like that. Oh, it's awful to see anything here. Well, the important thing is that we have here measures of the sign of intensity. In this case, we'll focus on concentration as a function of time. So we can follow here what happens with hexane, which is the end solvent. Almost nothing, and then a sudden increase. Virginomycin, the main factors of virginomycin are here. So it's interesting, we have a decrease in concentration, which indicates that nucleation begins in the evaporation phase. And then when we added hexane, we have a sudden decrease of concentration. The solvent itself, and here we have a minor factors of virginomycin, some other molecules that are together with the main factors. For the crystal analysis, what can we see here? Here we have the chord length, which is related to the crystal size, and here we have the counts, which are related to the number density of crystals with each chord length. So what we can observe here is that in the evaporation step, as time goes by, following time, we have basically a nucleation here. It's of course a secondary nucleation. We have already nuclei at the beginning, the start of this time shown here. And we have also some crystal growth here. It is interesting. When we add hexane, we have a rather different behavior. What we observe mainly is that the small crystals either grow or agglomerate. We have both phenomena occurring together. So this lowering of the concentrations at smaller chord lengths is followed by an increase in higher chord lengths. So as partial conclusions of this step, we observed nucleation in the evaporation step, crystal growth and agglomeration in the precipitation step. We saw that crystals grow up to a certain limit, and we know that further processing lowers the average crystal size. We have also seen the co-precipitation of other minor factors of virginism. So let's go then for the industrial process. Which factors that affect crystallization and may affect the final biological activity? The median temperature in evaporation, the concentration after evaporation, it means how much I will carry out how long I will carry out the evaporation, the stirring rate, of course it breaks crystals, it may break, the time for anti-solvent addition, and the ratio anti-solvent to solvent. Well, how can we do that in an industrial process? In academia, we usually frown upon this kind of approach, but it is important here. We can change things in an industrial process in a very narrow range. So the best thing that we can do is to make a statistical analysis. We can change temperature, concentration, stirring, anti-solvent, time, and solvent ratio in small steps, up and down. So actually, each line of this table is a different industrial batch. And we get here, as a response variable, the microbiological activity. And the statistical analysis is quite complicated, but anyway, we can get some optimized operation conditions with high temperatures in the evaporation step, lower anti-solvent to solvent ratio in the precipitation step, and a shorter addition time of anti-solvent in the precipitation step. And actually, by doing that, over 1 year of operation after this, the average biological activity of the product increased in 11%, and the incrustation was completely avoided. But, okay, this is the industrial side. We are in academia. Why does this happen? After all, crystals look very similar. We know the mechanism of action of these antibiotics. So we know how it works. What we don't know, and it's a hypothesis to be tested, is that one of these minor factors observed in precipitation may play an important role in the reaction. That means there may be either another synergy or inhibition. We don't know that. We just hypothesize that we have another mechanism taking place here. So the conclusions of this part, the first two are somehow redundant. So we have the crystal analysis and lab scale shed light on the industrial process, and the industrial process may benefit from what was learned in the laboratory investigation. And for us in academia, the new questions that arise from both the lab and the industrial results may be an interesting indication of the path to be followed. So I would like to end by thanking my former PhD students whose work was presented here, Erica, Kelly, Sebastian, and Rodrigo. My colleagues, Everson, Professor Gerd Maurer, Adriano Zoni, and André Bernardo. The Brazilian funding agencies, FAPESP, CAPES, and CNPq. The German funding agency, DFG, which also supported part of the work in Germany. Fibro Animal Health Corporation, Metler Toledo, which lent us the workstation. To the Atmos Group for the invitation, and to all of you for your attendance. So, if you have questions, I promise to do my best to try to answer them. So, thank you very much.
Thank you so much, Professor Pessôa. It's now time for questions. If you want to ask one, please enable your microphone. or write it down in the chat and we will read it. Our YouTube viewers can write it down also. Thank you so much. Yuri will be handling the questions from now on.
So who's going to be the first one?
Nobody. Everyone is hungry.
I start.
Go on, Fred.
Thanks, Pedro. Your system is really complicated because you have a thermodynamic complicated system, a kinetic problem, and a solid phase that is for itself complicated. I have no models and not good model for those. I wonder, I'd like to study more kinetic phenomena here in atoms. We are starting, well, it's still embryonic, but we are looking for more kinetic phenomena. And what kinetic model are you using for this so complex system?
Well, what actually happened is that the kinetics is so complex that we can't use a complex model for that. We don't have data for that. Usually, when we have this data for example, for chord length and as a function of time, as a function of chord populations, we have some very simple models which relate this size population to supersaturation, to other properties. But these are really very simple. Usually the experimental scattering of data is so high, there is so much uncertainty in that we can't really model that. Okay, we have, of course, for B22, we have the second virial coefficient, we have models for that. But even for the solubility, when you go to the solubility, the Kohn equation, which is used to model, is so simple, and even it doesn't work that properly. So in this case, there are models that do that, but it's really difficult to fit parameters. Kinetics is complicated. We should stick with thermodynamics, with equilibrium.
Yeah. Just like for complement about the information about second virial coefficient, osmotic second virial coefficient, to get an idea if it is crystallized or form amorphous phase. You have shown that you increase the constant, well, you decrease the second virial coefficient, it's more negative, but it's still crystal. We know that there is a windows for crystal when you talk about second virial coefficient, but you haven't seen this. Is it? If I understood?
Yes, that's correct. Actually, we have seen when we used the data at the crystallization window, it worked. We got crystals, but then we tried outside this window, and we got crystals as well. There are two things that may be going on here. One is that we have many methods to obtain the second virial coefficient. Self-interaction chromatography, we have light scattering, and the results of these experiments are not always the same. So we may have some uncertainty with which is the correct second virial coefficient. So we did our experiments are self-interaction chromatography experiments. So we matched some experimental data from literature, okay, that it was working, everything. but there is a theoretical problem behind that. And the second question is that the paper by George and Wilson, which has shown that relationship between the value of the second Fourier coefficient and the outcome of the operation is actually restricted to some proteins and some salts and some conditions. So I believe that it is an indication, but it can't be.
Sure.
It's not the universal truth. We can have. That's why we tried outside the crystallization window. We are researchers. We have to do that. And if we do more research in this aspect, I believe that we will probably get more data of crystal phases outside this crystallization window. But it's not a criticism to George Wilson. No, it's just it's more complex than that.
Thank you.
So, Professor Raja, would you like to ask your clarification now?
Can you hear me?
Go on, please.
So, Pedro, congratulations on your talk. It takes me back to my master's days of nucleation and growth rate and crystallization. So anyway, you use Kahn's relationships, Kahn and Hilliard. Why do you, this is more for spinodal decomposition, right? Spinodal decomposition, Kahn and Hilliard was proposed. That means there are alternating structures with fluctuations, right? So why do you use only Kahn and Hilliard? Why don't you go to, say, growth rate or nucleation kinetics?
Actually, this use of Kahn equation is what is
Why
usual.
don't you mention Hilliard also? There's a Hilliard guy also. Kahn and Hilliard.
Yeah, that is it's well known as Kahn equation. Actually, what happens here is that what I've shown with Kahn's equation is that how it's usually done. And I've seen people here in the Butantan Institute and showing things like that, and it's used. It's because it's a simple relationship just for the equilibrium curve. And so actually those experiments that I've shown we didn't try more sophisticated modeling because of all the experimental uncertainties and so on. The use of the Kahn equation was primarily to show what's the overall behavior, and to this audience here, I believe that many of those who are watching us here never heard of Kahn's equation. So I try to show how it is done, usually done, but it's more for an illustrative purpose than anything else.
Can I ask another question?
You're welcome.
I like your size distribution measurements in two ways of creating supersaturation. By solvent evaporation, you create a nucleation distribution, initial nuclear distribution, and slow growth. Then anti-solvent addition to promote growth instead of nucleation. This is basic idea, I imagine.
Yes.
The distributions are somewhat familiar, right? The Gaussian type, semi-Gaussian or Poisson type distribution in the first phase. The second phase is seems to be there is some lack of control of other factors, like you talked about encrustation. So that means that your size of your reactor somewhat small. There are some binding energy problems. The surface may be in play. Do you ever think of that? Things like, say, protein can bind to the surface of it or the interface. There's a binding energy and there's a reaction rate, right?
Right.
So what type of reactor you use, or your vessel is only glass?
It's only glass, yes.
Specialized 3506, or did you coat it with the anti-protein?
It's mainly glass. The vessel, the instrumented vessel is glass, In the industry, it's not glass, of course, it's stainless steel, but I don't recall here by heart which is the material. But in the lab scale experiment, it's glass. Of course, with the stirrer is a metal one, it's stainless steel or something like. Actually, the conditions in the lab scale are not exactly the same. We try to use the same as in the industrial process because we wanted to understand what's going on in a lab scale. We can instrument the vessel in a lab scale, but we cannot do that with this precision in the industry. But they are not exactly the same. Of course, we have to change the steering rate, we have to change many aspects. And one of the aspects is also the material of the vessel.
So you feel that the phase equilibrium approach with the liquid virial coefficient still have some possibilities of predicting incrustation and changes in nucleation rate and growth rate kinetics?
I'm not sure. Actually, we did.
It's
so complex.
Binding energy. Basically.
Yes. It's so complex that it's difficult to think on, because we know how to measure the second virial coefficient of proteins using many techniques. But in this case, we have the solid phase, the compound which is crystallizing is actually a mixture that is crystallizing. So even to define the second virial coefficient in this case, we don't know, for example, the cross second virial coefficient, how would it be, how it would affect the outcome. It's a very deep aspect to be considered.
So final question is, what do you think of the Schaffers who are trying to predict the amorphous precipitations and crystalline precipitation simultaneously with supersaturation? There are various papers, I mean, the last is maybe about 2015, Schaffer. I think there's a Netherlands group who's trying to predict for drugs the percentage of amorphous against percentage of crystallines growth with time, right? Using fluctuation theory only. See, there are many articles in Physical Review and things like this. So, like you have amorphous and crystal. You say outside the so-called crystallization rate you climb. Okay, I don't understand this crystallization rate yet, but anyway. Everywhere there is crystal, everywhere there is amorphous. Anyway, let us say that it depends on that size distribution, right? What is amorphous for me means, okay, it's a size distribution problem. So there are people trying to predict how much amorphous phase there, how much crystalline phase there in solids, precipitation rate. There are 2 or 3 types of mappings and theories. Did you have any comments about them?
For drugs, drugs are usually not so complex as proteins. It may be, but in any case, a solid phase of drugs are also really indeed complicated. We get not only crystals but different crystals depending on the situation. So with proteins, we don't have this problem. We just either we get an amorphous phase or we get a crystal. Sometimes you get some changes in the crystal habit and this kind of thing. For drugs, I've seen, but just briefly, I'm not aware in deep of how it's done. But I would say that it's very difficult to make such a map using just one parameter, because it depends on just the fact that we have our seeds in the medium changes the outcome of the. So we can get primary, secondary nucleation it's different, and so it's not just a parameter of the liquid phase which dictates the outcome of the solid phase. If I got your question right.
Thank you very much, Pedro. Just one more. Finally, I visited your lab. Maybe I don't remember 3 years ago or 4 years ago. You showed me your equipment for this. I don't remember who it was. Recently, you have made modification like single crystal detection growth, or what modifications you have made in your equipment. I think the first person or second person was doing doctorate at the time.
No, actually, this equipment here belongs to the Universidade Federal de São Carlos, Federal University of São Carlos. It is a collaboration with them. We don't have the equipment. Unfortunately, I would love to have this.
Okay, thank you.
You're welcome.
Do we have any more questions there?
May I make a question?
Yes, please.
Admiro? Yeah.
Please.
Pedro, a very nice presentation you have made. Following this, several experiments you have done to try to explain these different batches from industrial scale. You have 4 different batches, and you made several lab analyses and was not able to explain the difference in activity related to many variables, many analyses also. Have you seen about the possibility to have different solid phases that can change the activity, as you have shown before, that when you have the amorphous phase and others, the solid phase, the activity was quite different. Possibility to have different crystal phases that you have different activities as well?
Yes. In some cases, the different solid phases yield different activities, different biological activities. And it's in pharmaceutical industry, it's quite important because sometimes the industry try to first make a patent in one crystal phase and then in a second crystal phase to extend the patent. But in this case specifically, we made some X-ray diffraction experiments to check if we really got the same crystals or different crystals, and we didn't observe any different any really difference between these crystals. And there is another aspect. Usually the influence of the type of crystal in the biological activity is related, for example, in the bioavailability of the drug inside the bean. There's some questions like that. And in this case, the activity is measured in a very simple experiment. Let's measure the halo. We have a bacterial culture, but we put a solution. So it is already dissolved. So it may have a relationship with shelf life and so on. But in this case, the activity itself is measured after the dissolution of the antibiotics. So the problem that may happen with bioavailability and so on will not appear in this kind of experiment.
So you still have something to look at.
Yeah, yes. No, there are many aspects that are interesting. Solid phase is a terrible thing. It's nice to.
Thank you.
You're welcome.
More questions?
Would someone want to ask a last question before we finish?
Can I ask?
Sure.
Elvis here.
Elvis, open your camera.
My camera. Okay.
Yeah, it's nice.
I'm here.
Okay.
Thank you for the excellent seminar, Pedro. The topic is very interesting. I have a little question. Your X-ray diffraction can see the amorphous phase or not?
Yes. What happens is that if the solid phase is an amorphous one, we don't get those peaks. We just get a very flat mountain. Those sharp peaks that we observe in the graphics are indication of definite crystal phases. And it's the position of the position and the height of these peaks that indicate if we have the same crystal phase or different ones.
Yeah, but if you have a mixture of amorphous phase and crystal phase, and you do your Pounder X-ray diffraction, maybe you cannot see the amorphous phase in your X-ray.
You in this well, I'm not
because
sure.
you have you have a little fluctuations.
Yes.
Plus your
the overall.
Yes.
What happens specifically in this case is that we have from literature the crystal structure of one of the factors.
Okay.
Not both, because they're crystallized together. They crystallize together, but we can also, you can purchase them from Sigma Alders. They're extremely expensive, but from literature we have the S1 specifically. And what we observe is that we have the same signature, apart from some peaks, it's the same thing. And for the crystal S1, because in this case we have just one compound, one crystal. So the X-ray diagram is really from a crystalline phase.
Yeah. Okay. Nice. Thank you, Pedro. Thank you.
Yeah, well, I thank you for the question.
So, if there's no one else who wants to ask a question, I think it's time to wrap it up. So thanks for the discussion, people. And let us take a moment and thank again Professor Pedro for the marvelous presentation. Thank you.
I thank you, Yuri, Fred, and all the Atoms team. Thank you very much.
Thank you, Pedro.